Prokaryotic transcription is going to start with what we call initiation. And initiation is really all about the promoter region. So with initiation, the polymerase, and really we're talking about the holoenzyme, really needs to bind the promoter. And once it does bind the promoter, then we're really going to be ready to move into the next step. So holoenzyme binding the promoter, specifically binding the promoter at that -35 consensus site, is what we're going to be starting with. So once the holoenzyme has bound to the consensus sequence, this is really going to start the whole process. The sigma enzyme is going to leave very quickly after the holoenzyme has assembled on the promoter. But the role of this is very crucial because the sigma helps to recognize the consensus sequences. So if we didn't have the sigma enzyme, then we probably would not be binding in the proper place on the DNA. So it is important that it's there to get us started. However, once we do get assembled on the promoter, sigma is going to leave. And when it leaves, that will be the end of this first stage, which we're calling initiation. Another very important thing that's going to happen during initiation is that this consensus site is going to help facilitate strand separation. And when we say strand separation, we're going to actually see that the hydrogen bonds that are really in here, which I haven't shown in detail, they're going to be split. So if we were to look at that bubble, we're going to see that we do have the strands opened up, so that RNA polymerase is going to be able to actually look at the single-stranded DNA regions. We're not going to have, though, a really large section of the DNA ever opened at any one point in time. The second stage of transcription is going to be elongation. And elongation is going to begin as soon as that sigma component of the holoenzyme actually dissociates. So once we have the strands opened up a little bit, we do call this little area that we have the transcription bubble. So this transcription bubble will, for the most part, be covered up by the polymerase enzyme itself. And what this bubble is going to allow it to really look at the DNA sequence so that it can build an RNA copy right on top of it. So we're going to be using the bottom strand in this case. So we're using the strand that is 3' to 5' as a template. And that will be because we want to make our RNA 5' to 3'. And we always have to have anti-parallel synthesis. So when we start to make this, if we look at it, we're going to see that we actually build our new RNA right on top of this, and we'll actually see the end of the RNA molecule actually out here and single-stranded. So while it is underneath the enzyme in the transcription bubble, we will actually have the RNA, actually hydrogen bonds to the DNA template. And that's how the enzyme is going to know which bases to actually incorporate, because we're going to use the base pairing rules. Across from a C would go a G, and across from a T would go an A. If we are talking about actually making the RNA, we're going to put uracils in instead of thymine because it is RNA sequence. So this elongation process is going to continue with this transcription bubble moving as the RNA polymerase itself is actually moving down the strands. Now, RNA polymerase itself will actually be opening up that transcription bubble as it goes along, and as it goes along, the transcription bubble is also closing behind it. This whole process is going to take place until we get to a specific region of the DNA, which we're going to call the termination signal. So at some point on the DNA template, there will be a specific region, and I'll just put it right here, that will be recognized based on its sequence as being a termination signal. And again, once we hit that sequence, then the RNA polymerase is going to know that that really is as far as it needs to go. So for the termination part of this, it is going to happen when we hit or we run into the termination signal, and this is when the RNA synthesis is actually going to stop. And at that point, we will see everything really dissociate. There are two different ways that this happens in prokaryotic cells, and one of the methods is that we're going to be using a little protein that's called Rho, so RHO. And what this Rho protein does is it kind of follows along behind RNA polymerase all along. And here at the termination signal, there's going to be a sequence that slows that core enzyme down. So the core enzyme gets really slow, and really what happens is the Rho enzyme or the Rho protein ends up bumping into the core polymerase or the core enzyme because the core enzyme has been slowed down. And once it bumps into it, it really kind of just bumps everything apart. So that at that point, transcription stops. The enzyme itself, the core enzyme, is going to dissociate. The RNA molecule would be released, and the DNA sequences are going to actually close back up on each other. So that, since it involves the Rho enzyme, this process is what we call Rho-dependent termination. But we also have Rho-independent termination. And with Rho-independent termination, we're going to have regions of the RNA, which we're actually synthesizing, that fold up with themselves. And this structure that we get, we refer to it as a hairpin. And this is meant to be like a bobby pin you might be familiar with, that kind of folds like this. And since RNA is single-stranded, it can actually hydrogen bond with itself. And that's how we end up with those hairpin structures. What this is ultimately going to cause is this is going to cause the core enzyme to actually stall. And once it stalls, very shortly after that, everything is going to dissociate. And that would then be the end of the transcription process. That particular method is going to be called the Rho-independent termination.